In this video, we're going to look at the structure of RNases. And we're going to start with our group 1 ribozymes. And the group 1 introns are the most abundant self-splicing intron type. Remember in the previous video we talked about group 1 and group 2, so we're looking at group 1 here. And so far, we know of over 2,000 different types of group 1 ribozymes. Now, typically, group 1s will contain 9 different double helical segments, and we call these P1 through P9. And P just stands for base paired segment. Now, we divide these P's into two major domains that make up the catalytic core. That would be P4 through P6, and P3 and P9. So P4, P6, P3, P9. Now, if you look at this 2D image, it's really confusing to start with, but what's happening is we're trying to represent this 3D RNA on a 2D space. So this is our primary sequence. And you can see that we have these red lines. These red lines are just saying you have base pair interaction between the two different subunits. Now, also in this structure, we also have metal ions. Magnesium, they're usually there for structure stability and also the catalytic activity of these ribozymes. Now, if we focus on the X-ray structure a little more, we'll see that it has 3 stacks of aRNA-like helices. So aRNA is a helix that seems to be kind of flat. You can kind of see that a little bit in the green there. It's really hard to see in these images, but it's like a helix that's been squished on itself. Also important are this A-rich bulge and this tetraloop. What these do is that they have your bases sticking out, and you can really see it in the tetraloop here. See, our bases are kind of sticking out at the loop, and they're going to go and interact with different segments of our long arm here. Again, it's really easy to see in our tetraloop because we have a area that's called the tetraloop receptor. So the tetraloop kind of just sits in this tetraloop receptor, and you have base stacking here, and you have hydrogen bonds with uridines to help stabilize this structure. It's same thing's going on with the A-rich bulge as well. It's just harder to see with all this RNA. The next structure we're going to look at is tRNA. And tRNA is roughly 76 nucleotides with a lot of chemical modifications. E. coli has 60 different tRNA genes, and tRNAs will take this cloverleaf shape where you have 4 stems that have base pairing, and when you first transcribe it, the primary tRNA transcript, you'll have extra nucleotides at the 3' and 5' end. And so before this tRNA is ready to go, you have to excise and trim these extra nucleotides. And this process is a lot like the rRNA process we talked about in an earlier video. In fact, they will use some of the same nucleases to do that processing of your terminal ends. Now let's look at one of the ribozymes that is responsible for this, and that's RNase P. So RNase P is a ribozyme whose catalytic function is carried out by the RNA, not by the protein, but the RNA carries out the actual chemistry. And what RNase P does is that it'll process our RNAs and also modify the 5' ends of tRNA. So the protein is basic, which means it's positively charged, which makes sense because RNA is negatively charged. And what the protein really does is it just helps stabilize this pRNA and speeds up the reaction rate 2 to 3 times faster. So pRNA will work by itself as a catalyst, but this protein is really just help to stabilize. So it'll bind tRNA and it will also help release the product. Eukaryotic has 9 to 10 different proteins, so we're looking at a prokaryotic version right here. And this pRNA is like the other protein enzymes we've studied. It can do multiple reactions. not just one. So the self-splicing enzymes, they did one reaction and they were done. But p-RNA is a true enzyme. It can do many reactions. And if we look at the active site of the p-RNA, there's conserved U residues, and we also have two metal ions sitting there. What we know about DNA and RNA, they're probably magnesium, because that's the metal that often associates with DNA and RNA. And it's thought that these metal ions will help with catalysis. And there's a minimum portion of RNA that is always in all of our known RNases, and it's known as the universal minimum consensus structure. And the idea is whatever first p-RNA was around probably had this minimum structure, and then over the eons it evolved to be more and more complex, but it still had this minimal structure that was required to do the catalytic event. Now we're going to finish off by talking about post-transcriptional processing of our tRNA here, and we're actually looking at the tRNA that encodes for tyrosine. And a lot of eukaryotic primary tRNAs have introns and many extra nucleotides at the 5' and 3' end, which is the same for E. coli like we just talked about. The intron, interestingly enough, does not use a spliceosome, but we need an actual protein that uses ADP. So we have an ATP-dependent splicing endonuclease that will take care of our intron for us. Then we need to join our exons by ligase. So you can see tRNA is different than the rRNAs and mRNA introns we talked about earlier. At the 3' end of every tRNA is CCA. And this is where your amino acid will actually go on the end of the CCA. Now, in the primary transcript of eukaryotes, we don't have the CCA. That's what we're showing right here. So we need to add the CCA by a polymerase. And it will use CTP and ATP to add our new nucleotides here. Now for E. coli, the CCA is already in the gene. So we don't need an enzyme to do that. But E. coli do have the CCA adding polymerase. And the idea is, in case something went wrong with the 3' tail or it got damaged, this polymerase couldn't go and repair its function, because remember, we need the CCA to attach our nucleotide to. So if we don't have this, the tRNA is going to bind to the ribosome but be worthless because it's not giving a nucleotide away. With that, that's the end of this video. I will see you in the next one.